what sort of principles this is working off of, and it's the implications are pretty significant. I do ask a bit of housekeeping. I do ask, be aware that this stage will boost the boost anyone's voice to the whole crowd, so please stay off the stage, and please let only Physwolf or whoever's talking have the microphone, because it does the same thing. Other than that, Phys, please, if you may come to the stage and take it away. Thank you.
Do I also need the microphone, or can you hear me?
You don't need to hold it. It's just a prompt.
Okay. Put it over here. All right, hello everyone. I'm Physwolf. Today I'll be talking about some of my thesis work for my doctoral thesis. This is one of the modalities that we invented in this group. Most of this is around simulation work, though a lot of the precursors are based off of a lot of experimental stuff. And just to frame this, we never actually got around to building this. I'll talk about this at the end. Also, fun fact, you don't always get things right in science. This is a great example. There's nothing in here that we don't think won't work, but certainly some of the fundamentals I think we got wrong. But we'll touch on that as we get to it. First, I was asked to do like a little bit of a background about myself, just to frame that. A little bit about Physwolf, the physics wolf. I'm very original with names, if you couldn't tell. So first, my academic background. So I have a Bachelor of Science in Engineering Physics from the University of Alberta, which an engineering physics degree is a very broad thing, depends on the institution. In this institution, it's mostly like an electrical engineering degree, with a lot of focus on like device level physics, and then kind of at the same time you're doing a general physics degree, so you're covering topics of quantum, statistical, thermal, E&M, a lot of E&M actually, and solid state, all that kind of stuff. As well as you're taking a bunch of pretty advanced math courses as well, along with a lot of the other physics students. So it's just a giant mess. I followed that up with a PhD in biomedical engineering from the same university, which is a funny story. To ask me about it later. You're not really supposed to do a PhD at the same institution, but whatever. Screw what's expected. And this is mostly focused on biomedical imaging devices, primarily around ultrasound, photoacoustic systems, system architecture, image processing, these kind of things, everything that's related to these. Also, subnote, I'm not going to talk about it here, but man, ultrasound feels like one of those fields that's like 80 years old, and they're still just doing absolutely insane stuff in that field. And then I followed this up with a postdoctoral fellowship at the University of Waterloo for 4 years, where kind of continuing on my PhD work, not quite what I'm going to talk about today, but looking at novel absorption microscopy approaches in there. Also some cool work. Maybe talk about that another talk. People always talk about institutes, they never show pictures. University of Alberta, which is actually this really, really nice campus. It's on the river valley. It's got lots of those old like brick buildings. Kind of nice. University of Waterloo, exact opposite. It's all built from like the 60s. It's all these awful concrete buildings. Everything's crushed together. I mean, it's a good institution, but it's not the prettiest looking thing. And then the last kind of topic is my employment background, because I've also been employed for a while. I've been employed at a biomedical imaging startup aiming to commercialize the use of PARS. That's just the acronym, that photoacoustic or photon absorption remote sensing. So we were commercializing that. There's some cool stuff there. I'm like, oh right, videos won't play. That's right. Well, the right thing is a video, just showing some different contrasts that we're picking up. Here we're primarily looking at doing label-free histology. So these are taken essentially of like just unstained pieces of tissue and using absorption contrast to get at that cellular structure. My current role in my current company, which I won't be able to go very much into detail for obvious reasons, I'm currently acting as a lead optical systems developer, because of the engineer is a protective term in my jurisdiction. And we're looking at developing biomedical treatment and diagnostic systems for ophthalmic diseases, primarily looking at vitreoretinal diseases, so diseases inside the eye and on the retina. And my day-to-day task is managing a team of developers and acting as a technical expert on a broad amount of subjects for the company, which is always fun, because everyone always asks you questions that they need, precise answers to. Okay, so a little bit of primer. What is optical imaging? You've seen a microscope. You might have used a microscope. It's really advanced stuff, I know. any kind of imaging system relies off contrast, and the most kind of common contrast that you'll generally run across is scattering, like you got like a piece of rock or something. Transmission, you've heard this before. There's also absorption, and absorption is always kind of given a little bit of a a side place compared to the other two, though you're probably very familiar with an absorption microscopy technique. Anything that fluoresces, you've probably seen fluorescent microscopes, 2-photon, second harmonic. These are all these kind of things rely at least partly on absorption properties, so it's not commonly kind of considered that way. But that's not quite what we're talking about. So when we're talking about absorption microscopy, we're not talking about this. So you imagine like you feed light into a system, and if the system can fluoresce, you get kind of this radiative relaxation. It's going to shoot out photons. But you can also get it's still radiative. I don't know, my PI like to term this non-radiative, but it's really thermal radiative. Non-radiative in quotation marks. Relaxation, so thermal relaxation. This is the main one that we're talking about, and this is the really cool one, especially when you start talking about biological chromophores, like red blood cell, hemoglobin and red blood cells here. So you imagine we're specifically talking about, especially in this talk, that thermal relaxation. How do you image a thermal relaxation? That's kind of the caveat here, right, what we're after. And how you do that is called photoacoustics, or photon, or photothermal. So generally, normal photoacoustics, you send in like a pulse of light, and then that pulse of light gets absorbed by the chromophores that you're targeting, and some chromophores that you're not targeting, but won't worry about that right now. And then if you do that at a tight enough, fast enough pulse, you get a nice pressure, a thermal rise, a pressure rise, and then that can send out acoustic waves. And generally this is done with acoustic transducers. So it's a very common system would just be like a big pulse, like nanosecond laser, and like an ultrasound detection array. Though that's not really the best thing for it. But that's most of these kind of images are taken with something kind of like that. And it's great because you can get like really deep into the tissue because you're not limited by optical transport. You're limited by acoustic transport. So man, as long as you can get photons in there, you can go centimeters deep and get blood vessel structures. If you have a tumor that's labeled, or if it's a melanoma or something like that, that'll give you contrast. So it's really, really cool. But the general problem, especially when you start going really small and trying to look at really delicate things, is that most conventional photoacoustics, they need that acoustic part. It's kind of a critical thing, right? You need to have that acoustic contact with the sample, and it's not great when you're talking about something that's delicate, that you can't just smear an ultrasound gel and crush it under a transducer. And so this kind of brings up this whole field of non-contact photoacoustics, which is sort of the precursor to a lot of this work. And just to highlight, I don't need to really go through this. Basically, the field is bad. Okay, non-contact photoacoustics is awful. Trying to measure acoustic signals from a very small acoustic source across air into some kind of detector, it's awful. It doesn't work very well. And a couple of groups have tried and failed pretty miserably. This is where my initial research comes in, which was started by a 2-year senior in the graduate lab, and he would say he invented it, but I'm gonna put some caveats on there. But anyways, it came out of our lab, this photoacoustic remote sensing microscopy, or PARS. You're gonna see that acronym here and there. Here's a PARS system. Boo! Look at that terrible optical desk. It's beautiful. And essentially, this is a non-contact imaging modality. It gets the optical absorption contrast. We managed to get it with the fraction limited optical resolution. It has pretty high sensitivity. If you ever see an SNR on an imaging thing, just ignore it. It doesn't mean anything, because it's like you can define it however the hell you want. It's just a scam. But it does have pretty good sensitivity and pretty good contrast. You can see kind of an image. We'll look at some images on the other slide. It's relatively fast because you can go like single point pretty much as fast as you can pulse a laser, as long as you can detect these acoustic signals. We'll kind of talk about what that means. And you can kind of still go a little deep. That's not gonna be a fun talk for the future. We won't really touch on that here. So how does this work? Okay, so let's say you have a chromophore, this red blood cell. You send in a laser pulse, just like regular photoacoustics. Boom, that goes in there. You excite it. Look, it's excited. You built up pressure. Now, the main kind of effect that you're taking advantage of in PARS here is something called the elasto-optic effect, where this pressure essentially creates a refractive index profile. So the refractive index of a material, which is effectively like the optical impedance of the material, is generally based off of the density of the material, right? So anything that alters the density even slightly can change the refractive index. And the cool thing is, which actually might be another fun talk that'll be a lot heavier than this talk, is I did a lot of work on, a lot of my thesis work is actually on this detection stuff here. And essentially, because you have an interface, you have an optical interface, even if you have a very small modulation in the refractive index of one of those materials, you actually get a pretty huge amplification of the reflectivity off that interface. So you get this really kind of cool effect where even though you might only be changing that refractive index by like 0.002 or so, but it's actually fairly detectable against this kind of DC reflection that you're getting off the same area, as long as it's near an interface. So it's really cool. If that sounds interesting, we could do another talk on that sometime. It'll be way heavier than this, though. So you excite, you build up pressure. That pressure creates a refractive index modulation, and then you can see that refractive index modulation just with a regular continuous wave laser that's focused at the same exact spot. And you'll see this as a tiny little time varying oscillation. And that the magnitude of that little oscillation in just the intensity coming back, there's no interferometry or anything here, just in the intensity coming back, it tells you something about how much absorption happened, how much heat was generated. And this is like super non-trivial because this is a game of dynamic range. You have a really strong back reflection coming back, and you're trying to look at a very small modulation on there. So it's kind of a fun technical problem. Here's some pretty pictures. Here's an image of a mouse ear, essentially. So you put a mouse ear flat on there. The skin, everything's intact. The mouse doesn't have hair, but which helps. But bam, blood vessels, system works. It's great. It's a microscope. You can see really small things. There's some really crappy images of red blood cells. Why are they crappy? Mostly because our scanner was really crappy. And building microscopes is difficult beyond just the optics, if you want to take nice pictures. And again, like I really want to emphasize, these are pretty simple systems. Like you're sending in an excitation beam, and you have like a CW detection beam. It's basically just a simple punt probe. It's all about how you're using the excitation and what you're doing with monitoring the back reflected light. So like I alluded to before, we could do a whole other talk on the at least proposed physics behind this. I don't know, is it right? Who knows? It's all really difficult. Any kind of biology and photon tissue interaction gets really complicated really fast. The other thing I'm going to just do a really quick primer on that I cannot go in very much detail because it's kind of related to my current role. We also got to talk about coherence-gated, optical coherence tomography, which relies on coherence gating. OCT is really fascinating. Basically, it's taking advantage of low coherence interferometry to be able to tell like spatial, to give like spatial, what's the word I'm looking for? Be able to tell where things are in space. I'm spacing on the word. So, like, for example, the most common application of this is in ophthalmic imaging. So imaging the eye, the anterior and the posterior of the eye. Here's like an image of like someone's retina. And this is like a cross section. The really cool thing, and this is the main takeaway that I want you to get with OCT, is it allows you to get an entire depth scan at once. So you imagine when you're doing an image like this, you're not kind of like scanning left, right, and then going in depth like you would have to with like, say, a confocal microscope. You're just going like, I want this entire line, and this entire line in depth gets acquired at once. Then you move over, entire line acquired in depth, which is one of its big strong points. Okay, so what we're going to do in this talk is we're going to take this cool thing that's PARS and this cool thing that's OCT, and we're going to smash them together. Why do we want to do that? Because this is what a B scan of just some like small carbon fibers look like. So these are 7-micron carbon fibers. We're looking in depth and lateral, and they don't look very circular, do they? Right? Our axial resolution is pretty awful for a variety of reasons, which I won't get into. So there's this. It also takes a long time. Like, you know, if you want to image something in depth, or you want to image a volume, if you have to point scan every single point, man, even if you're going, you know, in the megahertz regime for your point rate, it just takes a long time to scan through all that, like a long time. And especially if you're trying to image biology does not like to stay still. So the faster you can go, the better. If we can do, you know, take advantage of that kind of coherence-gated detection, you know, we only have to scan in 2 dimensions to do a full volume instead of 3. Okay, so how does this work? So we're gonna move over to simulation space here. This is the hat we're gonna pretty much wear for the rest of the talk. And let's just build a little structure here. So let's say here's a profile in depth, and we have a couple of structures in here. Let's say we have some optical absorbers, these 2 little small ones. Let's say we have a top interface. Let's say it's like air, water. We have an absorber here, an absorber here. They also have, you know, a refractive index profile. If we excite this whole thing, then you can imagine we've created a pressure rise, and we've created a little perturbation in the refractive index. We're plotting the refractive index profile in depth. Refractive index profile in depth. We create that pressure rise at those absorbers, and then the pressure is going to flow away from those absorbers, right? Seems pretty straightforward. If we look at that in how an OCT would see this, so if you simulate how an OCT would see this whole process, if you just kind of look at the OCT itself throughout time, so now we have time and depth looking at this profile, and again you have, you know, the absorber and the absorber, these gray lines in here. It doesn't look like anything. Nothing's happened, right? But remember, this is a game of dynamic range. So let's just say we take all of these subsequent A scans and we subtract out the first one. And that's what we get here. So now you're looking at the difference in refractive index profile, the difference in the scattering profile in time. And now all of a sudden these absorbers light right up. Okay, that's interesting. That's interesting, right? This was a video, but it didn't occur to me that this wouldn't play. But imagine they're animated. Just imagine in your brains. Okay, so going to go to the next slide. There we go. Okay, so we say, all right, well, if, you know, these photoacoustic induced elasto-optic effects produce scattering modulations in the sample, that's what we were seeing, those scattering modulations, those red kind of V's coming out there, and specifically a spectrodomain OCT is able to capture these single-shot depth targets. It feels like we should be able to do this. It feels like if there's some kind of way that we can do this kind of trick with an OCT, we might be able to detect these modulations. So there's a couple technical problems with this. First of all, the spectral domain OCTs generally aren't quite probably fast enough. If you point down to this time scale, this is nanoseconds, right? So this is really quite quick. And generally, spectral domain OCTs only kind of run at about a megahertz kind of range, so we're a couple order magnitudes off. So we need to develop a fast OCT, which is fun. And so my proposition here is a pulsed detection OCT. Now, and specifically, you could use like a femtosecond for running this detection. Femtoseconds are actually pretty common to use for OCT sources in spectral domain, especially really high resolution. But they're not used directly. You take your femtosecond pulse, you run it through a dispersing fiber to basically just generate a wideband CW source, essentially. So the proposition here is you just don't broaden the pulse. You leave it as a short nanosecond or a short femtosecond pulse, and you do your detection kind of in a flash. Okay, so super high level, just to kind of recap before we go back. What we're saying is essentially we're going to take 2 OCT images. We're going to take 2 depth scattering profiles, one where we haven't excited, we haven't put in our excitation pulse, one where we have, and I've exaggerated here just to highlight, you know, the idea. And we take the difference between these 2 OCTs, and then that'll give us our absorption profile. Okay, that's really cool. Here's another fun diagram, just because it was a good diagram. I already had it, and I didn't want to make it. So essentially, you're taking the difference in these 2 OCTA scans, these two spectrograms, Man, I do wish I could do an OCT talk. The OCT talk would be fun. Okay, so this was actually when we proposed it. This is like a brand new modality, right? So this is not you know, because we had to you have to argue to the community that if you're making something new, if you're doing you know, any kind of device research, like specific device research, you know, it's one thing if you're you know, looking at new phenomena, or you're trying to you know, measure something new about you know, properties or something like that. If you're actually trying to make an argument to the scientific community that you're doing a new process, you have to sort of prove that you're not, you know, other things that are already existing. And so, like, you know, we're not all these other things. We're not OCT angiography. We're not utilizing, you know, phase-based tracking. This isn't visible OCT, which also can kind of get absorption, but you have to do a lot of modeling and assumptions for that. It's using an interferometer, but it's not this, you know, specific blend of interferometric photoacoustics, right? It's a very distinct thing. And okay, so let's simulate this, because that's about all we're going to get to. Is we need to construct a simulation for this. We want to have some kind of phantom where we can put absorbers inside of a background. We are going to simulate, you know, putting an excitation pulse through here. We have to simulate the acoustic propagation of all that. And then we have to kind of compare a before and after case. We have to find some way of converting this 3D, you know, mass of refractive index profile into something that we can actually run a realistic simulation on without having to do an optical simulation through this. Because we're gonna have to do this point to point, right? You know, we could just make this all, you know, one giant wave, like multi-physics wave simulation, but it would be very costly and expensive to run pretty much anything. So we're going to make some approximations here on the detection side of things. Here's some math. Essentially what we're doing in a bit more detail is we're simulating the pressure field, just using this is commonly known as the photoacoustic equation. It's just a pressure wave on the left-hand side with this heating driving term on the right-hand side. And I'm not that big on a simulation. I don't like to do anything too fancy, so we're just going to use some finite difference time domain, where we're basically just going to take that above right equation there. We're going to do a bunch of finite difference approximations to those derivatives, and then we're going to just solve for our next time step. You imagine we take all this kind of decomposition of that equation up there, and we're looking for that next time step one. I'm looking for it right here. That comes out of that time derivative, right? And then so you just rearrange this whole thing for that one time derivative, which gives you this other equation down here, and that's your update equation. So this is a description basically of how the previous two states of the pressure field and the current state and the previous two states of your heating field, how they all kind of link together and kind of keep propagating through there. Then we had to, so now you imagine you let this acoustic propagation go around, then you can calculate what the perturbation to the refractive index profile is, which is this is the elasto-optic side of things, the top equation here. So you have your unperturbed refractive index profile, this n naught, and you're going to perturb that by this delta n, which is this elasto-optic effect up here. So essentially, you have this relationship between the pressure and how that's going to perturb the refractive index, which gives us, let's say, n prime here. And this is, if you're a mathematician, please look away. This is terrible abusive notation. But I was forced to come up with some way of describing my algorithm in math. So don't look at it too much. But essentially, you imagine we have this refractive index profile, and now I need to run an optical simulation on this to figure out what my complex reflectivity spectrum is going to be coming back off this thing, so I can simulate an OCT. That's really difficult, and you have to run a lot of wavelengths through for a good simulation. You imagine you have to run, some 2,000 or so wavelengths through this in order to get a good OCT simulation. I'm not going to do 2,000 optical wave simulations per interrogation point. So we're making the assumption that, hey, my OCT is going to be a super low numerical aperture, so most of my wavefronts are going to be relatively planar when they're interacting with this, and I'm going to just convert this into essentially a one-dimensional problem, essentially just tracing wavefronts as they're going through the sample, and then along kind of those ISO wavefronts, we're just going to kind of do a weighted average based on the intensity profile of the detection beam, this is what this is trying to show. Essentially, you have your detection beam, this I int, which has some distribution. It's going to do this essentially weighted average across that to make this into a one-dimensional problem. The one-dimensional problem, I can throw something called the transfer matrix method at this. So this is actually a really cool method. Essentially, if you have a big stack, a refractive index stack, this allows you to essentially calculate what the reflection and transmission profile of that stack is. So this is useful, for example, if you're trying to develop coatings and stuff like that. But we're going to use it. Oh my God, where am I in my room? I'm in a corner. So essentially, we can take that one-dimensional refractive index profile, and we can get our complex reflectivity from that, which would be the R there. You don't take the absolute value. And you can do that for however many thousands of wavelengths you need to do your OCT simulation. And then that gives you your reflectivity from that, your reflectivity profile from that point. And you can kind of finish that. That gives you your OCT sinogram. Do your Fourier transform, you get your OCT A line. So that's all complicated. We did a bunch of those simulations. The other aspect of this is that was actually still very slow and doesn't provide a lot of quick kind of feedback. So I also developed this linear shift variant version of this problem, which we might be able to argue in some situations. You can replace this with a convolution, and it's a linear shift invariant system. But essentially, what you're doing is you're saying that I expect any kind of target source to act as a point source. I expect my final image to become a collection of these point sources, a linear collection of these point sources. That's that linear shift variant version. If it's the shift variant, if you expect that to be different per location, if it's the same per location, then you change this into a convolution, and the processing gets a lot more straightforward. How does this all look? Just to highlight this result from earlier. But if we have two chromophores like there, you can see here's the comparison between that 1D simulation, that big complicated OCT thing, and our more simple point spread function based thing. So they agree pretty well. Just for fun, there's a PARS PSF with the same numerical aperture, which looks awful, of course. Okay, so then let's do a more complicated simulation. We got this primer down. So now we'll construct some phantoms where we have a refractive index profile and an absorption profile, and we can see how well this performs. So here's kind of a trivial case where we have both scattering contrast and absorption contrast, and of course these show up in the OCT. You can see the walls. You can see kind of the structures in the OCT. Some of them are a little hard. And hey, they also show up really good as absorbers. In fact, if anything, they have much better contrast. That's kind of the goal here. Let's make it harder. So a lot of times in biology, you don't get the opportunity of having scattering contrast. A lot of the times your scattering contrast looks like this. If you try to image something with an OCT, it looks like this. It looks like a giant mess of scatter and pixel and speckle. But even here, bam, you can still recover the absorption profile because it's giving you that refractive index modulation. This is nothing but surface. Biology is nothing but surfaces, right? So there's surfaces everywhere to take advantage of that PARS effect, that PARS modulation, and we can still kind of recover these, which is really, really cool. We did a bunch of other simulations, which I think I'll mostly skip over for time because we're running out of time. But essentially, we did a run to check what the timing. we checked what the timing looks like it needed to be. So essentially, on that second interrogation, we do that first interrogation where we do like an OCT image or an OCT A line. Then we do a second one that's trying to immediately follow an excitation because you're trying to capture. Remember, that's that pressure, those red V's from the start. So the timing between the excitation and the interrogation, that feels really important. We did a quick study on it. Yeah, it looks like it matters a lot how you delay these things relative to each other. That's definitely an important thing if you're trying to build something like this. Another aspect is just like noise propagation and discretization. Like I don't know, can I get away with a 10-bit ADC for this? No, it'll look awful. You need dynamic range. So we did some studies on there to essentially figure out what kind of ADCs and such setups you'd even want to use there. We also did another cool thing. So one of the sort of hallmarks of photoacoustics, because it's imaging absorption, optical absorption, a lot of chromophores, even when they have very small differences, have very different absorption profiles. And to kind of highlight that, if you look at like oxy and deoxyhemoglobin, I don't know if any of you have ever seen on like one of the health apps on your phone, you can put your finger up to like a light or something like that, and it tells you your blood oxygen saturation. Or if you've been in a hospital and they clip that little thing to your finger and it gives you your blood oxygen saturation. It's working off this effect essentially, and it works really well in photoacoustics. You can map out blood oxygenation structure. You can map that out inside of like a living thing. So hey, does this seem like it's sensitive enough to pick up these small differences? Yeah, sure. And again, it's going to be another game of dynamic range. It's a game of dynamic range, squared. You can see the two individual exposures, and all we're doing here is we're exposing two different excitation wavelengths. So imagine we're trying to target, one spot here kind of thing, where that, difference between the two chromophores is appreciable, right? And then you run this through like just a simple linear system, and you can kind of solve what the local oxygenation would be. So really cool. Okay, so conclusion. We were probably wrong. Science is hard. So the thing we were probably wrong about, which we discovered in like later research, is that it's probably predominantly a photothermal effect, not a photoacoustic effect, which is actually really good news. So photoacoustics are what's driving that need for that really tight integration, that like several tens of nanoseconds integration. If it's photothermal, we have an order of magnitude more time, which makes things infinitely easier. So a lot of the later work on the PARS mechanism is suggesting that it's more so thermal based as opposed to acoustic based. There's certainly going to still be acoustics in there, but that's actually good news for this. We assume that said that so cool, still novel, might make it easier. The other thing is we never built this thing. We had a couple of projects try to start building it, but it's hard and it's expensive, right? So a couple of the kind of key components you need to make this work. You need a high frame rate line camera for that OCT. In order to make it, you don't necessarily need that to work, but you need that to make it viable as an imaging system. So, even getting like, 100 kilohertz kind of line rate cameras, which are out there, and they're not horrifically expensive. They're certainly expensive for a graduate lab budget. That requirement for that short pulse, so you need to provide at least a sub picosecond pulse to make this work well, and those aren't free either. The rest is imaging. Oh yeah, and then of course there's all the other components that you would need in just a normal OCT system. So all the interferometry parts, all the control systems, the PC software, et cetera, et cetera. It all gets really complicated pretty quickly when you start trying to actually piece these together. So for a variety of reasons, we never got around to building this. Not for a lack of me trying, but that's kind of cool. And there was a bunch of funding, which I felt like I should include there. That's mostly the talk.
Hell yeah.
Because of money.
We have questions.
Imagine clapping.
Yeah, questions. Do we have questions?
Smacking controllers.
Where's the microphone? I don't know if you want questions.
Hand off the microphone to whoever asked the question. I don't think.
Okay, so this is sort of more of a backgroundy one that you mentioned near the beginning of your talk. You talked about signal-to-noise ratio and how you can define that however you want, quote unquote. I'm just interested in what you meant by that, because I can imagine in like medical physics it's more complicated than say astro.
So I think the whole aspect of like image quality characterization is a trap. It's a trap of the subject because it really matters what you're looking at, what information you're trying to get at. Something that is useful for machine vision is not necessarily pretty to look at for a person. I think a good example of this is, you imagine if you just had like an image that's just like a black field and like a single white pixel. You have essentially infinite SNR, but it's not very helpful. What you really want is some kind of combination of low signal or of low noise relative to what you're looking at, just because you want some kind of clarity, but you also want good enough contrast. It doesn't really do you enough good to be able to see one thing, but you can't see one thing relative to another, which is usually what you care about, right? Like even if you kind of, I don't know if I'll go back too many slides. If you look at some of these like actual pictures or actual images, gee, even here. So, I mean, this one's probably been processed to absolute hell, but you imagine there's noise in the background here, right? And certainly you could call some of the signal, but what do you call signal? Is this signal? Is that signal? Because your SNR is going to be different if you pick this as your signal, or if you pick this as your signal, and then that's your noise, right? Likewise for your contrast, if you pick like a contrast to noise ratio, so it might be some difference of here versus some difference in here, over compared with the noise or something like that. It's a mess. Also, there's things that look like noise that aren't actually noise. So, and OCT is a great example of that. That's in speckle. You can even see that in some of these simulations where you have this background. This is not noise. This is speckle. So this is a consequence of the fact that you're doing interferometry and you're looking, in this case, at the real component of that interferometric result, right? It's not noise. But you can imagine in a real imaging system, you'd have this and you'd have noise on top of it. And so it gets really complicated really quickly to try to separate those things. And most of the time, people don't define how they're measuring it. Right, and in academia that feels like a mess, right? Like they spew out numbers. What does it mean? How was it calculated? What was their process? It's a trap.
All right, fair enough. Things I would never think about when we point a telescope at a single object.
Yeah, I know. More questions?
Anyone else?
Yeah, I have a question. Actually, I have two questions.
Whatever, but, like, can you hear me?
Yeah.
Hold on, dark eternal.
Oh, sorry, were there multiple?
So I'm working with computer graphics stuff,
Interface.
but
Interface.
Interface.
What?
Stop. Yeah. You don't have the microphone.
It's okay. I mean, it's okay.
Dark Eternal was first, though.
Yeah, like, anyways, index of like refractive index has no relation to the density or to like the optical, like that's different, right? Like with optics and like the mass of something, like because like when something is less dense, you get a different refraction, right?
Yeah. So refractive index is specifically based off
have different optical properties, like refractive indexes. So like how does that work?
That provides your scattering contrast. So refractive index is generally, traces its way back to susceptibility, like electric field susceptibility, right? Which is kind of more of a broader concept, essentially like how electric fields propagate through material. But the refractive index, which is probably better thought of as like this optical impedance, right? That's what gives you this kind of information, right? Like if at the top here we have like say air and then a surface, the reason that you see this surface is because there's a difference in the refractive index there, right? So there's a difference in impedance which causes reflection, right? It causes reflection transmission at that interface.
I think. Or is it cool?
And then changes in pretty much any kind of material property, like changes in the density, brought on either by like a pressure wave or a thermal wave, something like that coming through will alter the refractive index. Dark Eternal?
Sorry about that. Thank you so much. I just have two questions.
Mm-hmm.
I was just sort of, thinking about how you were saying, biological things are like
I'll keep
always
holding
on
the
the move,
microphone.
and it's, like, oh, sorry.
Right.
Um, I was I was hearing how you were saying, like biological things are like always on the move, and as a result, they are like hard to capture. But I can imagine that it's not possible for all kinds of biological living things. But if you could like isolate, like blood cells, and you would like cool them down, to like a very low temperature, would that actually increase, the accuracy of the imaging that you get with the techniques that you are using, or
so
does that, temperature doesn't matter?
The issue with biology, especially if you get down to single cell level, is they're really temperamental, right? You really can't change their environmental conditions much, like, at all. Otherwise they'll just die, and they're no longer a cell at that point. They're just sort of a mush of proteins and stuff, right? So you need to pretty much, especially single cells that are kind of just out in like a dish or something like that, you have to be really specific with their environmental conditions, unfortunately. And then if it's on a living, so like when I'm saying like a living thing, I'm usually referring to an actual like living, like, animal or person. And then you start to run into all kinds of other issues, like the fact they're breathing, they have a heart rate. And if you're trying to do microscopy, even the fact that their heart's pumping, even if you're just looking at like a capillary network, it still moves and it still pulsates.
Okay, so my secondary question would be, so like with the the PARS technique. So like in the old past, they would use like an X-ray, and that would damage the DNA, like of the living things. But is the like the PARS technique, with the laser, like exciting the cell, is that like a more safe technique than what they're like using, like X-ray?
I mean, by the old PARS, you're just talking about like ultraviolet excitation PARS. That's like it's like kind of like that background slide that was sort of way back here. Is that what you're referring to?
Yeah,
because we've never done it. We've done it.
It's like excited,
Yeah, because we've never done any work back here. Like, we've never done any work with X-rays. Like the lowest excitation that we've ever really done is like 250 nanometer. And so you're in ultraviolet, which certainly denatures larger molecules like DNA. But it's an extremely useful contrast to take advantage of. It's still far less invasive than physically having to cut tissue out and do standard like histopathology on it.
Okay, thank you so much for answering my question. I don't know who wants to go next. Okay, BTEX.
Does anybody else want a question first? Yeah, I'll take. I do have a question. Howdy. So I actually did read through a bit of your thesis, and photoacoustic resonance is like 100-year-old phenomenon. Like you were mentioning a moment ago, there were some other technologies that also make use of the of that foundational phenomena. Maybe in simplistic words, like I mean, it was partially answered previously. So it sounds like this is going to be a let's say it allows for more in vivo imaging at a higher resolution than before, or at least that's the hope of this research.
Yeah, exactly. Like specifically this modality would likely be well suited for trying to image smaller structures in that like living tissue, right? So for example, similar situations that OCT is applied in, so ophthalmic imaging, you're looking at anterior, posterior of the eye, or looking at diseases. Also in endoscopy, so OCT, the other main use for OCT right now is in endoscopy. So you can make really, really small imaging probes out of these that usually the whole actual system is external, but you just have kind of like essentially like a little fiber going down. And this is used for looking at plaque buildup and stuff in arteries and so on. It's also used by dermatologists, so to look at, even there's even lots of pioneering work on being able to diagnose, skin cancers, just looking at them with OCTs. So I think these kind of applications would be pretty, far up there.
I think it was part of your presentation, but do you have any pictures of like the actual equipment? I've had an MRI before, and it's a massive piece of equipment.
So this one specifically wasn't built, but do we have? There's an optical bench, at least from the graduate lab. And this has a standard PARS system on it, but it's honestly not even most of it. I mean, there's lots of supporting stuff on here. The actual microscope itself is just this kind of tiny little thing here. And you can imagine, for example, like to get those like mouse ear images, you have sort of a 3-axis stage that you're putting under here, and you're physically just scanning the mouse around under it. Some of these, and this is kind of a topic of how you have to implement these kind of things in graduate labs. These kind of images, these took like an hour to acquire, right? Because you're physically scanning the mouse. You imagine you strap them on to the stage, and they're anesthetized and so on, and you're physically scanning them, and you're kind of getting point by point by point by point. But this is a huge field of view. This is about 1 centimeter wide. So it's an absolutely massive image. The raw uncut ones, because these are cropped down to kind of hide the ugly parts, for publication. But this is essentially like a centimeter squared image that's captured at. I think we were doing the 2 micron steps or so. So it's an absolutely massive image that you're acquiring there.
Okay, thank you.
Cool.
Anyone else have questions?
This is way above my pay grade.
Okay. Physwolf, that was really awesome. Thank you very much for that. You are able to do the second session as well, right? Or
Yeah.
okay. Well, what is unusual about this meetup is that we're going to be transitioning to the STEM Tea Time VR immediately after this. So we will be normally during our meetups. We also try to do a actual meeting to actually talk to everybody as a part of it. So. Just unusually, this time we'll be joining STEM VR for that. We will have a second meetup, which will be in 6 hours, 45 minutes from now. That will be our later, more American-focused time zone session. So for now, we'll try to figure out how to transition to the STEM VR, so hold on tight for that. Seriously, Fizz, thank you very much. Is there anything else you want to insert about yourself or anything before, while you still have the stage? He does really cool stuff.
Do I really do really cool stuff? I don't need to. No. I also stream. I'm a DJ. I run a nightclub, DogHouse Rave Night. If you like music, come check those out. There, plug over.
It's good. I remember to enable your voice when you jump on stage and went on Twitch.
Oh yeah, that's already messed up. Don't worry, I can't.
Okay.
For next time, I guess.
Round of applause.
Yeah.
Thank you, Chris. That was really good.
Clapping.
Yeah. So
clapping.
For now, we're gonna try to transition this over to STEM VR. So hold on tight. The next step is to basically just talk to each other. So just do that until we figure